So now I will shortly introduce Professor Luisi, who is actually has a long number of years of activity. Actually, he was one of the co-founder, among many other things, he was one of the co-founder of the Institute of Polymers at the Polytechnic of Zurich. He is now at the University of Rome Free in Rome. He's actually active, and you will understand from the talk, is currently researching on synthetic biology and models of the origin of life, from this autopoietic perspective, which is a very stimulating one. He has wrote already many books, and I know he's actually currently working on one with Fritz of Capra that I very much look forward to. So I leave the floor to Professor Luisi.
Luisa Damiano, and let me remind you briefly what this term chemical synthetic biology, or more generally synthetic biology, implies. Somebody says that synthetic biology is nothing else than a new, more fashionable form of biochemical engineering. Partly is true, but there is a new focus on synthetic biology, and that is to the very ambitious goal of constructing alternative life forms in the laboratory. Alternative life form, or at least alternative biological structure, where alternative means that they are not existent as such on our Earth. And the term making new life must immediately be dimensionated by saying that we are still working at the level of unicellular microorganisms, and the new form of life takes place through genetic manipulation of the extant microbe and unicellular organism. What people do is to insert genetic component into extant microbes, and producing in this way new microorganisms which indeed do not exist in our Earth, and therefore they are new, they are novel. Why do we do that? We do that mostly for making new products, for example, new forms of energy in the form of methane, in the form of hydrogen, or new drugs or new industrial catalysts. This is the really engineering part of synthetic biology. I keep saying, however, that there are two souls of synthetic biology, and the second soul does not use genetic manipulation for a series of reasons why several people don't like to do genetic manipulation. But also, aside from that, there is a different kind of approach, no longer the engineering approach to build something having a design a priori, but rather addressing the nature, asking nature, why did you do that the way you did it and not in another way? Why this and not that? For example, nucleic acid have ribose instead of all have ribose, which is sugar at 5 term, not very stable, not very popular in nature, and you can ask the question, why did nature use ribose instead of the most common, most stable, most popular glucose? And or you can ask even question of the type, why is the banana yellow? And as silly as this question is, by asking this question and really exploring it, you would be obliged to redo all kinds of evolutionary chemistry that takes you to, this plant and then the contingency that make it one color instead of another. You can ask why protein have 20 amino acids instead of 15 or 35. Why we have the 4 bases, adenine, thymine, guanine, cytosine, only these 4 and not the other, and why this and not that. And the main point is that the chemical synthetic biology can help you to answer, or at least to tackle this question, in that you can make in the lab this alternative form and learn in the meantime why nature really wanted to do or had to do it in that way and not in another one. This is connected to a very important principle of biological and chemical evolution, the principle of contingency, which is really the main driving force for all the progression of complexity in nature. You see in this slide the sentence that most of the important biostructure ribosomes or all enzymes or all nucleic acids are not with us because they are the most stable compound. They don't form spontaneously as such. Lysozyme is not with us, chymotrypsin is not with us because they are formed spontaneously as the most stable compound. They are made through a long variety of steps, through the pathway of contingency, and which can be defined in this way. Contingency of several parameters is the coincidence of several parameters which are independent from one another, but that converge in a given point and space to produce a certain event. I give you a clear example. In the work of Stephen Jay Gould, who is one of the most the fiercest pioneer of the theory of contingency, and he, for example, in his beautiful book Wonderful Life, consider the point about 1 billion years ago when the world of the unicellular organism yielded the way to the world of multicellular organisms, the so-called Cambrian revolution. And he said this mysterious step or jump from the uni to the multicellular organism took place because, I am paraphrasing a little what he says, because of the contingency of a series of parameters, temperature, pressure, concentration, salinity, pH, the presence of sun, the presence of wind, and change only one of these parameters, his famous phrase, run the tape again, and this point could have taken, instead of 2 billion years, 12 or whatever, or it might have never happened. This is the very bitter and important conclusion of the theory of contingency, that all what we have on our Earth might have been different or might have not have happened at all. The tyranny of one parameter, as Jay Gould says. But let's see some examples. Why this and not that in nature, whereby chemical synthetic biology can help us synthesize that, the alternative, that nature didn't have till the time or some other reason to build. For example, let's consider the proteins of our life. There are very many. There are about 10 to the 12, 10 to the 14, so 1,000 of billion or 100,000 of billions different proteins on our Earth. Sounds like a very large number, but consider the following calculation, a question that we ask our students. If you have a chain of long 60 amino acid residues, in how many times, in how many ways can you build this 60 polypeptide, knowing that you have at your disposal in each position 20 different possibilities, 20 different amino acids. You can put 20 here, 20 here, 20 here, 20 different here, so that the number is 10 to the 70, which according to a young calculation by Einstein corresponds to all atoms in the universe. And then notice that the ratio between the possible, this theoretical calculated number, and the actual, it's an incredible ratio. It corresponds to 1 grain of sand, the actual, with respect to all sand of Sahara, so that we live with our 10 to the 12 proteins in a grain of sand with respect to the all Sahara. So the question is how have these few proteins been selected out? Who has chosen them? Maybe they are the product of determinism because they have special thermodynamic and physical properties such as stability folding, or maybe they have been chosen by chance, by contingency. And there is a way to build them, the new proteins, in the lab. What you do is you construct by random, Monte Carlo, a DNA sequence, and if you do so, you make a sequence long, let's say 150 randomly. The probability that by doing this piece of gene, you find something that is existing already in nature, the probability is 0, or this ratio 10 to the 14 divided 10 to the 70. So that when you do these genes randomly, you always find something non-existing, and you can insert these in the gene of a virus. These will produce a protein, so that at the end you can have proteins produced by your virus, proteins which are, we call them never born, because they don't exist in nature. You see here the point of synthetic biology, producing structure which are not present in nature, but they are alternatives to the extant ones. Those are the two collaborators which mostly did that in the time of Zurich and then in Rome. And they produce a library of 10 to the 8 of these different proteins, of which they analyze 100 or so, finding that over 20% of them, of these never-born proteins, were nicely folded, and some of them, once you have the primary structure, you can ask your Rosetta computer model to predict the three-dimensionality, as you can see here, and people in the field of protein chemistry recognize familiar figures, helices and beta sheets and gamma and beta term, etc. And of them, some of them, we did some spectroscopy, like this very nice protein with 2-tryptophan, or in some case, we did the circular dichroic spectra. Here, for example, the original protein. Here, the protein denatured with guanidinium chloride. The absorption stops here. You take away the guanidinium chloride by dialysis, and you obtain reversibly what you had before. So this never-born protein behaves as our extant protein, so that here the implication might be that since our extant proteins do not have anything special from the solubility, from the stability in terms of thermodynamics, from the folding and maybe binding, then they are not chosen deterministically, but they are the product of contingency, and therefore life is then a product of contingency. The implication that we, mankind, are here as due to the game of contingency, which is a kind of bitter pill to swallow. But let me show you another example, the one that Luisa Damiano was mentioning about cells. Modern cells are extremely complex. Here you see the spaccato part of the metabolism of Escherichia coli, where each point is a chemical compound, each line is a chemical reaction, each chemical reaction is catalyzed by one enzyme, a specific enzyme. So this is a three-dimensional maze of thousands and thousands of reactions, all depending one from the other. And but it is precisely this extreme complexity that elicits the question: must a biological cell be so complicated and so complex? That's a different thing. Can one not build a biological cell which is much easier, a basic one? Here comes the notion of the minimal cell, a cell containing the minimal and sufficient number of components to be defined as alive. This question has its validity also thinking of the origin of life, where, of course, you cannot imagine, you cannot conceive that the first cell were already something like that with 1,000 genes and 50,000 different molecular components inside. They must have come, we assume, from a gradual increase in complexity. So where was the point of starting? Maybe this notion of minimal cell. And for that we use liposomes as a container, which have the structure of all our biological membrane, this lipidic bilayer. And for that liposome, we some years ago proved that they are efficient models for biological cells, also because they can self-reproduce. Consider, for example, these very simple molecules. They are fatty acids, which, according to Diemer and collaborator of the University of Santa Cruz in California, are prebiotic molecules. And these fatty acids can form micelles at alkaline pH or vesicles at lower pH. And the only other thing our chemistry should know is that there are precursors like the anhydride, you see COCO, which are water insoluble, or the ester, water insoluble, whose hydrolysis, however, you break here and you break here, produce this acid, which, according to the pH, make micelle or vesicle. Now look what happens. You have the very simple apparatus. This is synthetic biology in the simplest possible way because it's also prebiotic kind of chemistry, where you have a basic solution, natrium hydroxide, and here the water-insoluble precursor we have mentioned before, for example, the ester of the caprylic acid. Because of this basic nature here, at the macroscopic interface, you have an hydrolysis producing the acid, which at the so-called critical micelle concentration produce micelle, which are going to go here. So from here to here, you make micelle. Look what happens. You measure the caprylate being formed as a function of time. You wait for many, many hours, and there is a very low, sluggish increase of the caprylate concentration. But at this point, there is a jump, there is an explosion. All caprylate is formed and immediately transformed in micelle, you see the blue square, which are formed within seconds until then the reaction finished, because you finish whatever was your rate limiting component. What happens is an autopoietic process. Luisa Damiano mentioned that autopoiesis is a process which comes from within the boundary. You see, you form micelle at a certain concentration. Micelle can be seen in water as a little droplet of oil solubilized by the hydrophilic head group. These droplets of oil solubilize quickly the ethyl caprylate, which is water insoluble, is here. This is immediately hydrolyzed, producing new micelle, but more micelle you produce, more micelle again you produce because you hydrolyze more ethyl caprylate. This is an autocatalytic, autopoietic process which results in the spontaneous formation of vesicle, cell-like structure. Those are micelle. You can do the same with vesicle. So that we proposed at that time, we were already in 92, that this could be the process, an autopoietic process by which the first cellular component could be made. And you can even see micrograph showing the reproduction of these things. Now, going back to this minimal cell, here are the two main heroes of this work, Pasquale Stano and Teresa Pereira de Souza. Here is missing Professor Alfred Farr, who has been the host of all our electronic microscopy in Vienna. Thanks to him, we did the work, otherwise we could not have done it. The idea then is the following: to put extant DNA or nucleic acid in general, an enzyme inside one of these liposomes. And how do you put it inside? First of all, the idea is to put the minimal amount possible as to make a cell, not 1,000, but 2, 3, 4, and 5, 10, see what happens. There are several ways to encapsulate this red macromolecule inside. The simplest, conceptually simplest possible way is to inject a salt or a lipid which spontaneously makes liposome, and by closing they interpolate whatever is in solution. For example, you can do that with ferritin, which is a protein containing many iron atoms, so that you can see each protein due to this high electrodensity as a single point. and you can ask your student to, measure the frequency of ferritin as a function of the vesicle before and after certain experiments that involve, for the good Natalie Berklatz, the analysis of 1,000 micrographs. With this method, we did also PCR in liposome, and also still in long ago, 95 self-replication of RNA inside self-replicating vesicles, good exercise of really synthetic biology, self-replication of RNA. 20 years before, some modern group has done the same recently. And finally, at the end of the century, we were able to put the entire ribosome machinery inside the liposome, thus giving it the possibility of producing proteins inside the liposome. This was the beginning of an explosion, and several groups in the last few years have been introducing plasmid, mostly the plasmid of green fluorescent protein inside liposome to produce the protein. I like to mention the group of Yomo, a very bright worker in Japan, but also the group in Chicago, Noiro, Nakatani. Now, we work with a system developed by Ueda and collaborator containing only 37 enzymes and a total of 90 macromolecular components. Is still a large number, but still with respect to the 1,000 genes and the 50,000 components of Escherichia coli, we are already at a very low level. And this is what happens, that when you have this system and the plasmid for the green fluorescent protein inside the liposome, this becomes green because the protein is produced. So we have liposomes, model of cell. We make the most important function of cell, producing protein inside themselves. We have worked also with liposome as small as 100 nanometer radius, which you cannot see by confocal microscopy, but you measure the fluorescence in bulk. And now I tell you something in the last 2-3 minutes, which is going to be a small contradiction, because I, as leader of this group, I couldn't believe that this beautiful green experiment would happen on the basis of the following consideration. You see, we do entrapment in this way, easy with one component. Now, when you have 4 different components, the probability of having all 4 components inside one become very small. When instead of 4 you have 97, and there must be all 97 inside to produce the protein, because this is the minimal system, then I begin to think this. How can you put 97 components? The statistic is such that there should be the Poisson distribution, and if you have liposome with 100 nanometers, the probability according to the Poisson is absolutely 0. So the theory tells you that you should not have a result, but the results are there. So then clearly the theory must be not applicable in this case. This is an interesting point in science, when theory says it should not happen, and the experiment say, yes, it happens. And then we decide to study the statistic of entrapment using our friend ferritin, and we find something extraordinary, where indeed we show that the Poisson statistic does not work. And we found the following, that all material concentrate in only 0.1% of liposome, and most of the liposome are empty. We have in first approximation all or nothing, and which was very surprising. In overcrowding, together with empty ones, those are ribosomes, so not only ferritin but also other components. And actually, what happens when you plot all the data we have at our disposal is a kind of power law, which goes on, where you can have very many ferritin, and the probability 0.1% may appear very small, but if you have an Avogadro number of liposomes, 0.1% is several billion. And I conclude indicating that this kind of approach of synthetic biology, chemical synthetic biology, originated by the autopoiesis approach, brought us, maybe for serendipity, to a new law, perhaps of cellular biochemistry, perhaps to the origin of metabolism. Why so? Because if you think of the origin of life, when you have a solution with very dilute component, no reaction because they are too diluted, you add your lipid, which make liposome, then they are attractor, they concentrate, and reaction starts. So this is one way to go toward the final goal, which is synthesizing life, which will happen certainly in the next few years. And to those who ask, when do you do that? I can only answer with this kind of joke, where the young monk asks the master how far it is, and the master answers, hold your tongue and walk. Thank you very much.
Okay, thank you very much for your great talk. Actually, it raises many questions, but I don't know if we have some from the lecture hall. So maybe we are a bit late. So again, if you have a question, because you were today, you might feel a bit burdened by the mass of stimulation. So please contact us, and we will be happy to forward to the speaker.